Hi, my name is Marissa Hall, and I am a junior REU student in Dr. Charlie Martin's lab. And I spent this summer looking at invasive species in the bay and how the fauna that live within them react to artificial light. So the invasive species that I was looking at is this one right here. It's called Myrophyllum spicatum, or Eurasian milfoil. And as you can see, it's very structurally complex. This one plant has multiple branches coming off of it, and then those branches have more branches coming off, and then those branches have leaves, and then those leaves have other parts. So it is very structurally complex, especially in comparison to its native counterpart, which is this one right here, Vallisneria americana, which is coined the name tape grass, which should give you everything you need to know about its complexity. And so when looking at both of these and thinking about the animals that live within them and their exposure to artificial light, one can assume that the milfoil, the supposedly bad invasive species, would actually offer more protection from the artificial light that they're being exposed to a lot more due to urbanization. And so what we did to be able to kind of test and prove that is we went out into Mobile Bay, into Chocolata Bay, and we deployed a bunch of these lights right here. We floated them at the top of the surface of the water, left them out for about 6 hours, and then went back and essentially took a mesh bag core of the entire plant, brought it back to the lab, and we picked through all of the plants and took out all of the fauna that were once living and then frozen and then dead now that were in them, and we quantified them, and we also standardized them to the biomass found in each plant sample that we took. And so this is what we found. Right here is an MDS plot, and the first thing that you notice right off the bat is that there's a very clear discrepancy between the milfoil and the Vallisneria, with the milfoil being the green stars and the Vallisneria being the purple diamonds. There is zero overlap, and all of the milfoil is also clustered together a lot. The second thing that you notice is that there was no difference between the light and the no light samples that we took. All of it is very mixed in with each other between the two different species that we were looking at. And so when looking at that, you kind of wonder, okay, so we know that they're different, but what's causing that difference? So that brings us over to this chart right here. We have the total abundance of each of the samples that we took quantified out. And as you can see, the milfoil has a much higher abundance than the Vallisneria does, but there is not a significant difference between the light and the no light. And just to preface, for all of these graphs on here, all of the green is representative of milfoil and all the purple is representative of Vallisneria. And so when looking at this, yes, as I said, there is a more abundance of organisms in the milfoil than the Vallisneria. But when looking at the community as a whole, we see that the Vallisneria is actually a lot more even. It's a lot more evenly dispersed than the milfoil. So when looking at that, you're like, okay, milfoil has a lot more, but it's obviously being made up of fewer animals. And so we kind of began to wonder what is making up these discrepancies. And what we get is these graphs over here. And the main perpetrator of all of the difference that we see is an amphipod called Gammarus, which is also kind of crawling around on my poster up here. There were hundreds of them in all of the samples that we took, especially in the milfoil samples. And to look a little bit closer into that, we did a simpler analysis to get the average dissimilarity between the two different types of SAV. And we found that there's a 60% dissimilarity between the milfoil and the Vallisneria. And the Gammarus accounted for 53% of that, which is absolutely crazy. The next highest one is Diptera, which is fly larvae, and that only accounted for 8%. So when looking at this overall, we kind of learned that one, short-term exposure to artificial light during the day does not really affect the community assemblages between the milfoil and the Vallisneria, which makes sense. There's not really much time for them to have a change in their behavior. Their food, the algae that they eat, doesn't really have a chance to have a change in their behavior, and so that makes sense. But there is a big difference between what we see in the Vallisneria and what we see in the milfoil. And I hypothesize moving forward, as urbanization continues to bring us into the future and more fauna are being exposed to artificial light, milfoil can actually play a positive role in the lives of the little microscopic creatures that live within the bay. So that is all I have.